Hello and welcome to this GCSE Biology mini-masterclass. In this session I'll be covering all of the key concepts associated with enzymes, what they are, how they work, factors that affect enzyme activity before moving on to the required or core practical on this topic. That being a required or core practical for a number of different examples such as AQA or edX. So as always, there'll be plenty of exam practice including some modelling from me and plenty of examples for you to have a go at home. Let's get cracking. So again, if you've watched any of my previous videos, you'll know I use the bug technique to help interpret the exam questions. A quick reminder then, B stands for box or brackets, the command term, putting a spotlight on what the question is actually asking us to do. U, underline any key information in the question, even better use a highlighter pen. And G, doing a little glance back, essentially a proofread of our answer before moving on to the next question in the exam. Enzymes then, what are they? Well, enzymes are made of protein, okay? They're incredibly small, approximately 3 to 7 nanometers, which we would require an electron microscope to see. We've got an example of an electron microscope image here on the left-hand side. Now, you've probably been taught about enzyme action using little diagrams like this, almost like cartoon images. And the image on the left is just to show you that actually these little cartoon images are pretty close to what enzymes actually look like, okay? I'll be looking at the structures of an enzyme in a moment, but you can see here from this image that if you trace the outline of our orangey red enzyme here, there's this special little region in the middle, almost looks like a mouth, and that's the active site that we'll refer to shortly. Equivalent to this part of the cartoon image of the enzyme. Being proteins, okay, proteins of which are long chains of smaller molecules called monomers, that makes them a polymer. And those smaller monomer molecules that make up the polymer protein is amino acids. Okay, we need to remember that for our exam. How do they work then? Well, they catalyze chemical reactions. In other words, they speed them up without being used up themselves. They're specific, so they work on certain substrates, okay? The substrate being the molecule that locks into the enzyme's active site, okay? And the active site is said to be complementary to the shape of the substrate. In other words, they fit perfectly together. The active site, as I've just said, will lock onto the substrate. In some examples of enzymes, being those that break down large molecules, you would get one substrate locking into the active site of the enzyme, which might be broken down into 2 or more products. Some enzymes, though, are builders, such as those that help in the building of DNA molecules during DNA replication. And you would expect to get 2 or more smaller substrates locking into the active site to produce one bigger one. And importantly, as mentioned previously, the enzyme is not used up in that reaction. It will remain at the end, okay? And that links to its definition as a biological catalyst. Factors that affect their action then. Well, to begin with then, little link to chemistry, collision theory. Think of the enzyme and the substrate in this case. Those are molecules that must collide together. So the substrate needs to collide with the active site of the enzyme. Now, this little cartoon obviously shows the pair of them having eyes. They don't actually have eyes. They can't see each other. So they are moving around randomly. And we are obviously hoping they collide together for a reaction to occur. Now, there are different factors or conditions that can affect the likelihood in which the two collide together. Remember, when looking at the factors that affect enzyme action, the less time taken or needed to produce the product, the faster the reaction has occurred, okay? A lot of students mistakenly take a lower time, because it's a smaller number, as a slower rate of reaction. Think about running a race, okay? The less time you complete the 100-meter sprint in, the faster you have run, okay? So if we're looking at time, the lower, the faster. Enzyme concentration, if you increase that, will initially increase the likelihood of collisions because you've got more enzyme molecules. Increasing the substrate concentration Well, again, if you have a higher concentration of substrate, you've got more substrate molecules, again, more likely to collide. We've got a little graph here to go alongside the example of increasing substrate concentration. You can see substrate concentration increasing on the x-axis, and initially the rate of reaction increases. Remember, initially means to start with. It then starts to curve, slowing down until the line becomes horizontal, okay? It's plateauing. And the reason that's going to occur is your enzymes and their active sites are going to become saturated, okay? So you're eventually going to run out of available enzymes, even though you're adding more and more substrate, okay? So the rate of reaction becomes constant at that point. A final condition to be aware of at this stage is increasing the temperature, which will initially increase the likelihood of collisions and more high energy collisions. Why? Because the molecules, the enzymes and the substrate molecules have more kinetic energy. So they're moving more quickly, increasing the likelihood of high energy collisions. A couple of graphs to go with temperature then that we'll look at in more detail in a moment. Initially, we can see that as temperature increases, the rate of reaction is increasing. Don't forget, this one is referring to time taken for the digestion of our substrate being starch. So as the time is decreasing, that's clearly showing an increase in rate of reaction at those lower temperatures, increasing from 0 to about 38 to 40 degrees centigrade there. The graph on the right, again for temperature, showing the same thing. However, we've got rate of reaction on our y-axis. We can see the increase there. We'll talk about why there is this rapid decrease in the rate of reaction at much higher temperatures in a moment. The optimum then, we need to be aware of what the optimum means and how to identify it on the graph. Optimum meaning the best condition for enzymes to work. And that's normally when the conditions are perfect for the active site. Okay, their active site is in perfect condition to allow it to lock onto our substrate. How to identify the optimum from a graph? Well, again, we need to consider whether we're looking at a graph with time taken on the y-axis or rate on the y-axis. For this one, we've got time taken. The lower the time, the faster the rate has been. So I'm going to draw on my graph. That is highly advisable for you to do the same when provided with a graph in your exam. Draw all over it to help you answer the question. And I've picked the peak of the curve, the lowest point of the dip in this case, of this inverted curve, and that's in line with 40 degrees centigrade. So 40 degrees centigrade is the optimum condition. If I were asked to identify the time taken or deduce the time taken for starch to be digested at the optimum temperature, well, I can see the optimum is 40 degrees. I'd read across to the y-axis, and my answer in this case would be 3 minutes. Okay, 2, 4, 6, 8, 10. It's 1.5 squares up, so between 2 and 4 would be 3 minutes. And another example then, if I had rate on the y-axis, I'd once again draw on the graph to help me select the optimum condition, optimum temperature, and if I had to deduce the rate of reaction at the optimum, again, I'd read across by drawing a line across from the peak to the y-axis. Now, we've talked about the optimum conditions when the enzyme works best. What about conditions that cause a change to the structure of the enzyme and therefore a significant decrease in their activity? Well, examples are extreme temperature, very high temperatures, and extreme pH. So it could be a pH that is very different to the usual conditions of the enzyme. Okay, that might be very acidic or very alkaline, for example. In these extreme conditions, the shape of the active site changes. Okay, we say the enzyme has become denatured when that happens. And therefore, the substrate, the molecule the enzyme is trying to lock onto, does not fit. Essentially, the active site is no longer complementary to the shape of the substrate, and therefore the enzyme can't perform its job, and activity will rapidly decrease, and therefore you'd see a rapid decline in the rate of reaction. Well, what about the required or core practical then? The most likely experiment you've performed yourselves or had demonstrated to you in the lab is investigating the effect of pH on the rate of reaction of a carbohydrase enzyme, so one that breaks down carbohydrates, called amylase. And the carbohydrate that amylase breaks down is starch. Okay, that would be the substrate. Now, you might have used slightly different apparatus yourself. I'm going to talk through the most common equipment or apparatus used to perform this investigation in this example. We'd have a number of different test tubes, okay, and into those test tubes we would add a controlled volume of buffer solution. Remember, volume is the quantity of a liquid or solution, and our buffer solution has a known pH, so we might have one at pH 4, one at pH 5, one at pH 6, and so on. We'd add to that buffer solution a controlled volume and concentration of our enzyme solution, so that's our amylase, and a controlled volume and concentration of our starch solution, that's our substrate. And what we want to work out is how pH affects the time taken for the enzymes, which are represented by my little green shapes here, to break down the starch, which is represented by my little red squares in this image. So I'm trying to work out how long it takes for all of the red squares to disappear, basically. And what we would do is have one test tube with this setup, but for different pHs. That would be our independent variable in this scenario. Now, to test for the presence of starch, we would use iodine solution, and we would add iodine solution, which is an orange or yellow brown colour. We'd add a couple of drops to the dimples of spotting tiles, and I'd have one spotting tile set up next to one test tube, let's say at pH 5. I'd have another spotting tile set up next to my test tube at pH 6, another at pH 7, and then I've just got one at pH 10 here in this example. I'd add my few drops of iodine to those dimples, as I mentioned previously, and all of these would be orange to begin with. Now, at the very start, to test for the presence of starch, I'd remove a drop from the solution in my test tube and add it to my iodine, and I'd expect the iodine to go black to prove that starch is present. And I'd expect that at the beginning because the enzymes haven't had time to start breaking down the starch. What I would then do is every 30 seconds, I'd remove a drop from my solution and then add it to another dimple containing my iodine. And again, if the iodine changes from orange to black, I know starch is still present in my solution. When I get to a point that I've tested my iodine, I've added my solution to my iodine and the iodine doesn't turn black, in other words, it stays orange, I know that my enzyme has finished breaking down the starch. So starch is no longer present, okay? And what we might have to do is, from a results table, determine the optimum condition, so the optimum pH based on the results. What I'd like you to have a little think about at home is, looking at these results here, what the optimum pH was for our amylase enzyme. Pause the video and I'll review in a moment. Right, so we were looking at when the solution of iodine no longer changes to black when we've tested for the presence of starch. In other words, it stayed orange. So it can't be pH 10 because our iodine has gone black right the way through, which means starch is present from the beginning to the end. Therefore, the enzyme's probably denatured in this alkaline solution. Of the remaining three, we can see pH 7 only required 60 seconds for the amylase to break down the starch, whereas the others took longer based on the results. So pH 7 would have been the optimum. Some exam questions then. I'm going to model this first set before giving you a chance to have a go at some yourselves. Again, I'll be using that bug technique to help. Now, my first question asks me to identify what the independent variable is from this investigation. Quick recap on variables. Remember, the independent variable is the one that's been deliberately changed to see what effect it has on the results. And a little tip for you, it's normally the last step in the method that they give you in an exam. It will say repeat using different blah, blah, blah. That's your independent variable. If I was asked about the dependent variable, that's what you're going to measure and record to determine what effect the independent variable has had. So that's what you're going to write down in your results table if you're doing an experiment in the lab. And control variables are ones that you would need to keep the same, okay, to avoid them having any effect on the results. So I can see here the final step of my method is telling me that the experiment here has been repeated using a different pH of amylase solution. So my answer that for the independent variable, the one that's been deliberately changed, is the pH. The bit in bold there is essential. Next question. I've got to describe how the student would know when all the starch has been digested. Okay, they've told me they're testing for the presence of starch in this experiment, but they haven't told me what they're using. You've got to know what chemical is used to test for the presence of starch and what colours are associated with it. Now, if I'm having to describe when all the starch has been digested, I've got to reference iodine and it not turning black. Or I could say iodine would stay orange or yellow-brown. And finally, in this third question related to the graph, I've got to select what the optimum pH is for amylase. Now, I've got to look at the y-axis to help me do this. A lot of students make the mistake of just picking the highest point on the graph. No, no, no. The y-axis has time taken to digest all the starch on it. The lower the time, the faster it's been. I'm going to draw on my graph to help me. And I've got to pick out what this value is on the x-axis. It's 8 little squares above 6. There are 10 squares between 6 and 7, which means every little square is worth 0.1. Therefore, 8 squares above 6 is going to be 6.8. Jobs are good at. Right then, a couple for you to have a go at home. As it's the first you do question, I've provided you a little bit of support in the form of a labelled diagram needed for part 2 of these questions. Give yourself no more than 4 minutes when you pause the video and I'll review in a moment. Right then, the first one, why did the contents of both test tubes turn blue-black at the beginning? Well, they've both got starch in them. Simple as that. Next, explain the results of this investigation after 10 minutes. Now, the only difference between the two test tubes was the liquid from the mouth being in test tube 1 and the liquid from the stomach being in test tube 2, along with the starch and the iodine. We can see from the results that in test tube 1, the starch was broken down. That gets you a mark. In test tube 2, the starch had not been broken down. 2 marks so far. The most challenging part of this question was why. It was an explain question, and you're expected to know that the liquid from your mouth, your saliva, contains amylase, which is the enzyme that breaks down starch. However, the liquid from the stomach does not contain amylase, so the starch would not be broken down. That's what you needed for your 3rd mark. Couple more then for you to have a go at. Slightly different scenario here. Again, pausing that video, 3 minutes maximum. That's a mark a minute, and I'll review in a moment. Answers then. Well, for your control variables, these are factors that would need to be kept the same to avoid them affecting the results. Any 2 from that list there. Okay, you'll notice that temperature is not on the list because clearly in the final step, that's what's being changed. Okay, that's the independent variable. And for the next question, why the student left the starch solution and amylase solution for 5 minutes before mixing them, that was to allow them to reach the same temperature as the water in the beaker that they're sat in. 1 mark for that. A final set of questions for you to have a go at home then, these being a continuation of the ones we looked at previously. We've got 6 marks up for grabs here, 1 question being a 5 marker, so I've given you a mark a minute plus 1. 7 minutes in total. Pause the video and I'll review in a moment. Right then, so they're timing how long it takes iodine to stay yellow-brown. In other words, they'll know when it's yellow-brown, starch is no longer present, okay? And from our results then, we can see that as the temperature increases up to 35 degrees centigrade, the time taken is decreasing, so the enzyme activity is increasing, okay? As temperature increases above 35 degrees centigrade, the time is increasing and therefore the activity must be decreasing. Okay, you need to reference that very important temperature, 35 degrees centigrade, at least once as part of your answer. Whole lot needed there for 1 mark. Final question, explaining the results at 5 degrees centigrade and 80 degrees centigrade. Well, in both cases, the iodine did not become yellow-brown. We can assume it's stayed black. So, starch had not been broken down. Why? Well, at 5 degrees centigrade, which is obviously a very low temperature, the enzyme or amylase or molecules have got less energy. So there'll be less collisions, or you could say less enzyme substrate complexes are formed. Okay, that's the combination of the enzyme and the substrate when they lock together. 3 marks so far. What about 80 degrees? Well, again, it's not become yellow. The iodine has not become yellow. In this case, though, it's not to do with the energy of the molecules. It's to do with the fact that the enzyme or amylase is denatured, or you could have said the active site has changed shape, and therefore the starch will no longer fit. And that's a wrap. I hope you found that video useful. If so, please hit that like button, and for more Science Mini Masterclasses, you can subscribe to my channel. I'll see you in the next one.